So today we're going to talk about the membrane, and here we've got the guy from The Office, and he's like, insane in the membrane. Correct, because we have the fluid mosaic model, which suggests the plasma membranes are full of integral and peripheral proteins, glycolipids, glycoproteins, cholesterols, and most importantly, we have these phospholipids, which form the semi-permeable bilayer. Very, very insane. And the plasma membrane you're going to see is very, very insane, and it's a massive focus of both Unit 1 biology and Unit 3 biology, so I'd be paying attention extra closely. And why it's such a focus is because any contact that the cell, which is the basic building block of life, any contact that the cell has with anything outside of the cell has to happen through the membrane. All right, so it becomes sort of like the communication strand of the cell. Okay. In Nelson Bio, we're still in chapter 1, but page 24 to 43, so to the end of chapter 1. So we're going to do the 3 quick questions a little bit differently. Just, I want you guys pausing the video here and having a go at going through the 3 quick questions. So pause it now. All right, so hopefully you've done the 3 quick questions, and I'm actually going to go through them in the video itself so that you can have a look. If you have still some questions, ask me about them in class. But the first question asks, what's the similarity and the difference between prokaryotic and eukaryotic cells? On your test, I found this was actually quite an issue for some people. I'd say a similarity would be probably to do with that they both have genetic material. So they have that DNA to make proteins. That's probably one of the biggest similarities. Another similarity, so with the DNA to make proteins, they have ribosomes as well, and that's another similarity. But another important similarity is the presence of the plasma membrane. And there's a cytoplasm, obviously. They're probably the 2 biggest ones. There are other ones. Difference is now we can list heaps and heaps. The biggest difference, though, is the lack in prokaryotic and the presence in eukaryotic of these guys. Membrane bound organelles. That is the biggest difference. All right. Size is also a big difference. What else? Another big difference. The structure of the genetic material. So in prokaryotic, we've got linear chromosomes. In eukaryotic, we have circular chromosomes. So circular versus linear chromosomes. So quite a few differences. All right, so for question 2, we're looking at this little picture here, and hopefully you recognize it, because what type of cell is this? We've actually got an animal cell, and it actually looks quite a bit like your cheek cell, so it's actually an epithelial animal cell, which just means it's a skin type of cell. Okay, what is this dark stained structure in its center? Hopefully you know that's our nucleus. Cool. And then over here we've got an electron micrograph of the organelle responsible for photosynthesis in cells. All right. What is the name of this organelle? Hopefully you recognize it as the chloroplast. And what specific type of cell is it found in? It is found in plant cells. Why only plant cells? Because plant cells are the ones that do photosynthesis, right? And that's what the function of the chloroplast is. All right, let's go on to today's work. So we're in the second part of the study design. We're talking about crossing the plasma membrane. All right, there's another dot point in this, but for today we're going to look at the first dot point, which is the characteristics of the plasma membranes as a semi-permeable, so it doesn't allow everything in, boundary between the internal environment and the external environment of a cell. I want you guys learning to call the internal environment of a cell the intracellular environment, and the external environment, so that's the internal, and the external we're going to call the extracellular environment. 2 really key words, okay? All right, so what's the structure of the plasma membrane? The plasma membrane, which is also known as the cell membrane, it's a structural organelle. I don't love calling it organelle, but often it is categorized as an organelle. And what it does is it encloses all of the contents of a cell, and it acts as a division between this intracellular environment, so inside the cell, and this extracellular space outside the cell. Right? It's about 7 to 9 nanometers thick. Usually we can't see it with the light microscope. All right, and it's what we call a phospholipid bilayer. As you can see here, we've got, if you look here, we've got yellow heads, green tails, so that's one layer. All right, and then here we've got green tails and yellow heads. We've got 2 layers, so it's a bilayer. And these are phospholipids, by the way. And we've got all these associated proteins. We've got some carbohydrates over here. We've got cholesterol, which is a lipid, and it's living in there. And we call this the fluid mosaic model. Why? Because the plasma membrane is fluid, all right? It's constantly moving like this, all right? It's not a structure just that's rigid, all right? And it's also a mosaic. So if you were to imagine a mosaic is this thing that's made up of this glass, and embedded in that glass is all this different colored glass that gives it that mosaic structure. So here you can see we have that fluid mosaic model. You can more clearly see the bilayer of the phospholipids. All right, so there's one layer. And there's another layer, all right? And then interspersed, we've got these cholesterol rings, and these are crack that you guys are doing, all right? But we've also got some integral proteins, which are both of these, some carbohydrate or sugar side chains, all right? So you can see we've got this fluid structure, but we've also got a mosaic, right? We've got all these things interspersed inside. All right, so let's have a look at these phospholipids a little bit closer, because we have this really important bilayer of phospholipids, and really they're the biggest component of the plasma membrane. But what are they? They're made up of what we call a hydrophobic. And if we break down the word hydrophobic, hydro means water. Phobic is like the word phobia, which means to be scared of. So it's a fatty acid tail, which is scared of water, right? And that's this part here that we can see. And we've also got a hydrophilic, or a fatty water loving, that's what the word philic means loving, head. So it's a water loving head, okay? If those of you that do chemistry, you'd see the words non-polar and polar, all right? Whenever you see the word polar in biochemistry, that means that water is going to be added. So that's what this head is all about. If you see the word non-polar, it means water is going to be repelled, okay? So that's why we've got this structure where we've got the heads facing out and the tails facing in, because both outside the cell and inside the cell we've got water, and so inside the we don't really have the water, so we've got these hydrophobic tails. All right, so these are the proteins that are related to the plasma membrane. We've got two different groups of proteins. We either have the integral proteins or we've got the peripheral proteins, okay? It's really simply where they're found. That's what makes them integral. Integral proteins take up the whole membrane, and the peripheral proteins are just either on the outside or on the inside of the plasma membrane, okay? All right, so what's the functions of the plasma membrane in general, okay? The most important function, I would say, of the plasma membrane, especially when you're studying biology, is its semi-permeable nature, okay? So it permits the selective control of molecules either into the cell or out of the cell, okay? Take the cell, and it's going to separate the cell from its surroundings, so making that really clear intracellular, extracellular environment, okay? The fluid mosaic structure allows for flexibility in changing shape with cholesterol intertwined in the tails. That cholesterol actually adds to the flexibility, and it actually resists temperature changes, okay? So this is really important if the cell needs to squeeze into different spaces, or if it needs to be an exact shape for its function. We've also got proteins that are attached to carbohydrate groups. So if we had a little to this carbohydrate group, and they act as like a recognition marker. So if something bound to there for another cell, for example, that cell would recognize, hey, this is my own body cell, or hey, this is a foreign cell. Okay, and we've also got receptor proteins that will actually receive signals from the environment to tell the cell to do stuff or stop doing something. Okay, so it's very much like the plasma membrane.